So today let's talk about spectroscopy. So in this spectroscopy, what is the spectroscopy? Spectroscopy is the study of absorption or emission of this electromagnetic radiations by a matter. For example, if I'm taking a matter, I will expose this matter to a electromagnetic radiation. The energy which is obtained from this electromagnetic radiation should be either it should absorb it or it should emit it. So this spectroscopy is nothing but absorption or emission of this electromagnetic radiations through a matter or by a matter. The absorption or emissions of this EMR, we can call this electromagnetic radiations as EMR, will start from gamma region to up to radio region. So let's see the types of spectroscopy. So before going into types of spectroscopy, we will be having different types of radiations. The radiations are UV or visible radiations and infrared radiations, microwave radiations and radio waves and X-rays and gamma rays. So let's get into the types of spectroscopy. The spectroscopy is divided into three types. First one is based on whether the study is made at atomic or molecular level. And the next one is based on absorption or emission of EMR. And the third one is based on whether the study is at electronic or magnetic level. So let's say the first one, which is based on whether the study is made at atomic or molecular level. So let's see at atomic level. So this atomic spectroscopy, here the energy changes takes place at atomic level. That's why it's called as atomic spectroscopy. So the examples of this atomic spectroscopy is atomic absorption spectroscopy and flame photometry. Next one is molecular spectroscopy. In this, the energy changes takes place at molecular level. That's why it's called as molecular spectroscopy. So here the examples of this molecular spectroscopy will be UV or visible spectroscopy, and next one is infrared spectroscopy and fluorimetry. So let's get into the next one, which is based on absorption or emission of EMR, which is electromagnetic radiation. So let's say the absorption spectroscopy here. The absorption spectroscopy is nothing but here the radiation is observed. That's why it's called as observed spectroscopy. And here we will study about this observed radiations only. So if we are passing any radiation through any matter, I already told you that it will take that radiation. Okay, so we are studying about that radiation which is observed. We are not studying about the radiation which is emitted. That's why it's called as absorption spectroscopy. And the examples of this absorption spectroscopy will be UV visible spectroscopy and IR spectroscopy and nuclear magnetic resonance spectroscopy, which is called as NMR spectroscopy. Next one is emission spectroscopy. In this emission spectroscopy, when radiation is passed through a matter, some of the radiation is observed and some of the radiation is emitted. So now we will study about the emitted radiation. That's called as emission spectroscopy. And the examples of this are flame photometry and fluorimetry. Let's get into the next one, which is based on whether the study is at electronic or magnetic level. So let's see the first one, which is electronic spectroscopy. In this, the study is done using electromagnetic radiation without the influence of magnetic field. So the examples of this are UV visible spectroscopy. So next, see the next one, which is magnetic spectroscopy. In this, previously we are not using any magnetic field, but here we will use magnetic field. So the study is done using electromagnetic radiations under the influence of this magnetic field. That's why it's called as magnetic spectroscopy. So the examples of this are NMR spectroscopy. So let's get into the UV spectroscopy, UV visible spectroscopy. So the first one is UV or visible spectroscopy. In this, we will study about the absorption of this UV radiation. So, as we are talking about the radiations, like electromagnetic radiations, here we will see how much of this UV radiation or visible radiations have observed. So, the range of this UV radiations will be from 200 nanometers to 400 nanometers. And when it comes to visible radiations, 400 nanometers to 800 nanometers. So the compounds which are colorless will be observed under this UV radiations, okay, UV range. And the one which have colored, they are observed under visible range. So let's see how this will work, how the equipment will work through a video. So this is the UV spectroscopy instrumentation. So let's see how this instrumentation along with how this will work. Okay, and this is an animated video.
Hello friends, today we are going to understand working and instrumentation of UV visible spectrophotometer. So first coming to the radiation source. So what is radiation source in UV visible spectrophotometer? Generally we are using combination of hydrogen deuterium lamp with tungsten filament lamp. So whenever we are using hydrogen deuterium lamp, then it will produce the wavelength of 200 to 375 nanometer, and when it is combined with tungsten filament lamp, then tungsten filament lamp is used to produce the wavelength 375 to 800 nanometer. But entire range of tungsten filament lamp is 350 to 7500 nanometer, but here we are using only up to 800 nanometer. So these are the radiation source which is used in UV visible spectrophotometer. Now this radiation will come to the next step and it will go to the monochromator. Monochromator is the combination of entrance slit, lens, then gratings or prism, then exit slit. So this radiation will enter into the monochromator through entrance slit, then it will go to the lens, and when this radiation, which is polychromatic radiation, will pass through the lens, then this lens will focus the radiation into the gratings, or we can use prism also. So when it will go to the grating will convert this radiation into 7 parts or 7 different colors, which is Vibgyor, and then these radiations which is Vibgyor goes towards the exit slit like this. So this is the complete setup of monochromator. Now coming to the complete details about monochromator. So first entrance slit will be there. So through entrance slit, all the polychromatic radiation or radiations coming from the radiation source will pass through the entrance slit and it will go to the lens. And here we are using collimating lens. That lens will focus the radiation towards the gratings. And what is gratings? Gratings is an alternative of prism, and it is advanced version of prism, because in the surface of gratings many small tiny prism are present. In 1 mm area there are 1200 to 1400 small grooves like structure are there, and that grooves or each grooves acts as a prism. If we are not using grating, then we have to use prism. Prism is also doing same thing, means main work of prism or grating is that it will convert the radiation into 7 parts, which is Vibgyor. Now this Vibgyor will go to the exit slit, and from exit slit we can select optimum radiation, and then this radiation will go through the chopper. This is the chopper. What is the function of chopper? Chopper will convert the radiation into 2 equal parts like this. Then this radiation will go to the mirror. Then these mirrors will reflect the radiation into the next level. In next level there will be sample and there will be reference. Okay, so one radiation will pass through the reference and one radiation will pass through the sample. Okay, so for sample or reference holding, here we have to use cuvette, and that cuvette is made up of quartz. If we are using cuvette of quartz, then it will cover the entire range of UV visible, because Quartz will not absorb UV light as well as it will not absorb visible light. But if we are using the Q8 of glass, then it can be used only in visible range because glass will absorb the UV light. Now this radiation will pass through the Q8 and it will go to the next level. After going to the next level, this radiation will go to the detector, and there are two detectors because it is double beam UV spectrophotometer. So first reference one will go to the D1 detector 1 and the sample radiation will go to the detector 2. So there are three types of detectors used in UV visible spectrophotometer. First one is photomultiplier tube. In short, it is known as PMT. So what is photomultiplier tube? So when radiation which is passed through sample or reference, it will come to the detector. If we are using photomultiplier tube, then radiation contains photon and that photon will go to the photomultiplier tube. Then it will be converted in the form of electrons, and that electrons will go to the dynode because series of diodes are present in photomultiplier tube, and the function of diode is that it will multiply the electrons. So those electrons will be multiplied and ultimately multiplication of electrons takes place. From one photon, approximate 10 to the power 6 electrons can be generated. Now coming to the second type of detector which is used in UV visible spectrophotometer. So that is photodiode array. It is also known as PDA detector. So how PDA works? In PDA detector or in photodiode array, there are various diodes are present, and these diodes will convert electromagnetic radiation or UV visible radiation into the form of electrical signal. And this PDA detector covers entire range of UV visible, that is 200 to 800 nanometer, and generally this PDA detector is used in HPLC. Now coming to the third detector which is used in UV visible spectroscopy is charged coupled detector. It is also known as CCD. So in CCD, silicon surface will be there, and when radiation will come to this silicon surface, photons will attack on the surface of silicon. Then these photons will be converted in the form of digital signal by using CCD or charged coupled detector. So these are various detectors which can be used in UV visible spectrophotometer. So now in next step, this detector will pass the signal towards the amplifier. So amplifier will amplify the signal, and this amplified signal will go to the recorder, and ultimately recorder will record the signal in the form of UV visible spectra. Spectra will be like this. So this is the working and the instrumentation of double beam UV visible spectrophotometer.
So till now we have seen the ultraviolet spectroscopy and visible spectroscopy, instrumentation and how it will work. So let's get into IR spectroscopy. In this IR spectroscopy, it will give valuable information about the structure of the compound. Why we are using this? To see the structure, to get the information about the structure of the compound. That's why it is also known as vibrational spectroscopy. When a molecule get exposed to infrared radiation, it will result in vibrational transition. That's why it is also called as vibrational spectroscopy. So let's get into the video how this is working and what is the instrumentation.
In this animation, I am going to explain about instrumentation and working of IR spectrometer. IR instrument is constructed by radiation source, mirrors like M1 and M1 dash, reference cell, sample cell, beam chopper, mirror M2 and M2 dash, again mirror M3, slit S1, diffraction grating, slit S2, detector, amplifier, and the last one is recorder. Now coming to the details of various parts of infrared instrument. In this animation, I will discuss the construction and working of dispersive infrared instrument. Fourier transform infrared or FTIR instrument will be discussed in the upcoming animations. First part of IR instrument is radiation source. It may be glober or nichrome wire on a ceramic support, and they are electrically heated from 1200 to 2000 degrees Celsius to produce IR radiations. Next parts are mirrors M1 and M1 dash to reflect the IR radiation towards the reference and a sample. Reference holder cell generally contains KBr because it is transparent throughout the IR range, which is 4000 to 400 centimeter inverse. Now coming to the sample holder cell, which contains sample to be analyzed, and the next part is beam chopper, which is operated by a motor and alternatively sends radiations from reference and sample respectively towards mirror M2, M2 dash, and finally mirror M3. Next part is slit S1, and then coming to the diffraction grating to improve the resolution of the IR spectrum. Next part of IR instrument is slit S2 and then detector. Various types of detectors may be used in IR instrument, and these detectors may be bolometer, thermocouple, thermistor, Gole detector, photoconductivity detector, and a semiconductor detector. Next part is amplifier to amplify the signal, and the last part is recorder. Now coming to the working of IR spectrometer. So first we will start the radiation source, and due to electrical heating it will produce IR radiations. These radiations will go towards the mirror M, which reflects half of the radiations towards M1 and half of the radiation towards M1 dash. Now M1 will reflect the radiation towards reference cell, and the radiation will pass from the reference material KBr without decreasing in the intensity of IR radiation. At the same time, mirror M1 dash will reflect the radiation towards sample cell. Some radiation will pass from the sample and some radiation will be absorbed by the sample. So intensity of the passed radiation will be decreased. When the radiation reaches to the beam chopper, it will alternatively send the reference and the sample radiations towards mirrors M2, M2 dash, and finally M3. So first I am taking the reference radiation for explanation. These mirrors will reflect the respective radiations towards the slit S1, and then it goes to the diffraction grating. Grating will diffract the radiation, so resolution will be improved, and this diffracted radiation will be passed through slit S2. Slit S2 will allow the radiations only in the range of 4000 to 400 centimeter inverse. These reference radiation will reach to the detector, and the detector will analyze the intensity of the radiation. Now chopper will send the sample radiation. At that time reference radiation will not reach to the detector. So sample radiation will also follow the same steps and it will reach to the detector like this. And this alternate process will be continued throughout the analysis. Finally, detector determines which frequencies have been absorbed by the sample and which are unaffected. Intensity of the absorbed radiation will be decreased and the signal will be amplified by the amplifier like this. And finally, IR spectrum will be visible in the recorder. In infrared spectroscopy, spectrum will appear in terms of percent transmittance versus wave number. For complete detail about IR spectrometer, you can watch my dedicated video.
So now we have seen IR spectroscopy instrumentation and how it is working. So let's get into the applications of IR spectroscopy, why we will use this IR spectroscopy. We will use this in identification of various functional groups and in structural elucidation. As we know, now only we have seen. To get the structure elucidations, we will use this one. And next one is identification of drug substance by comparing the IV spectra of the substance under the investigation with that of standard identification of the drug substance can be performed. Like we can compare the standard and the drug sample. Okay, and it is also used in identification of impurities of the drug. So when we are taking any drug substance, it will contain so much of, as it is chemical in nature, it will consist of so many impurities. So when we insert this one, it will also, as we have seen in the video, it will also show the impurities peak also, so that we can delete that one and we can proceed with the pure one. So now let's get into the mass spectroscopy. So this mass spectroscopy is used to determine the molecular weight. Okay, mass. That's why it's called as mass. So to determine the molecular weight of a compound. It is also called as positive ion spectroscopy or line spectroscopy. So let's see the video how this mass spectroscopy will act like.
Today I am going to explain about instrumentation and the working of mass spectrometer through animated video. Now coming to the different parts of mass spectrometer. So first one is slit A, second one is slit B, third one is slit C, fourth one is slit D, fifth one is slit E, sixth one is detector, seventh one is amplifier, eighth one is recorder. Now coming to the various chambers present in mass spectrometer. So first chamber is sample vaporization chamber. It is present between slit A and slit B. Second chamber is ionization chamber. It is present between slit B and slit C. Third chamber is ion acceleration chamber. It is present between slit C and slit D. Fourth one is mass analyzer. It is present between slit D and slit E. Now coming to the first chamber. So here sample will be inserted, and in second chamber, that is sample ionization chamber. In this chamber electron gun will be present in the form of tungsten filament. In this chamber electron collector will also be present, which will collect the electrons. Now coming to the third chamber, it contains various electrodes. First electrode is supplied with positive potential. Second electrode is supplied with negative potential. Third electrode is supplied with more negative potential, and so on. So it will accelerate the ions. Now coming to the fourth one, that is mass analyzer. So in the mass analyzer, magnets will be present and they will form magnetic field, and due to that magnetic field, deflection of the ions takes place. So this is all about instrumentation of mass spectrometer. Now coming to the working of mass spectrometer. So first sample will be inserted through slit A into the sample vaporization chamber, and here if sample is present in solid or in liquid form, then it will convert in the form of vapor due to heating, and the heating is given by heating coil. Now this vaporized sample will go further through slit B and it will enter into the ionization chamber. Now electron gun will produce the beam of electron, and that will strike the molecule, and due to this electron beam, molecule will lose one electron, and ultimately it will convert into the form of positive ion, and this positive ion is known as radical cation or molecular ion. Here energy produced by electron beam will be approximate 70 electron volt, and the ionization potential for the molecule will be approximate 10 electron volt. So due to high energy, M plus ion will convert in the form of fragment ion also, that will be M1 plus or M2 plus, etc. Now these positively charged ions will go towards the slit C, and their positively charged electrode potential will be there, and due to this positively charged electrode potential, these positive ions will be repelled in the right side, because in the right side negative potential will be there, and ultimately they will get some velocity, and with that velocity they will enter into the mass analyzer through slit B. And the low molecular weight compounds will be deflected high, so path length will be higher. So ultimately large molecular ions will reach to the detector first, then a smaller molecule will reach to the detector, and ultimately at the last smallest molecule will reach to the detector. Now these molecules will be detected by the detector, and here photomultiplier tube is used as a detector, and ultimately this signal will be transferred towards the amplifier. Amplifier will amplify these signals, and finally it will be recorded in the recorder, and we will get the mass spectrum. So this is the complete explanation about mass spectrometer and its working. For more details you can watch my video lecture on instrumentation of mass spectrometer. Link is given in the description.
So now let's get into the applications. The first one is structural elucidation and impurity detection. So let's get into this structural elucidation. By using this, we will study the fragmentation pattern, peak matching, and percentage of abundance of isotopes, and we will also see the structure of this compound. That's why we will use this mass spectroscopy. And impurities detection. Already I told you in NMR, when we insert this compound, it will not only show the pure compound, it will also detect the impurities, like it will show the peak of this impurities. When we get to know the peaks of this impurities, we can easily remove them. We can easily separate them. Let's get into the NMR spectroscopy. NMR is nothing but nuclear magnetic resonance spectroscopy. So let's see how it is working.
Explain about instrumentation and working of NMR spectroscopy. NMR instrument is constructed by compiling different parts which are radio frequency generator, magnet, sample holder, sweep generator, receiver coil, amplifier, detector, and a recorder. It is also known as oscilloscope. Now coming to the working of this NMR instrument. So first we will put the sample in the sample holder, and the sample must be present in the form of solution. If we are talking about 1H NMR or proton NMR, so in that condition sample must contain protons or set of protons. Now in next step, radio frequency generator will generate the radio frequency in terms of megahertz, and due to these radio frequency or electromagnetic radiation, all protons will be aligned in the direction of this radio frequency, and the direction of this radio frequency will be perpendicular to the receiver coil. In this animation I am discussing about constant radio frequency NMR instrument. Now coming to the next step. So here magnets are present, and these magnets will generate strong magnetic field, and this magnetic field is controlled by sweep generator by the help of Helmholtz coils. So these coils can control the magnetic field, means they can increase or decrease the magnetic field. If we are increasing the magnetic field, then resolution will be better, and the direction of this magnetic field is perpendicular to the radio frequency. Now in specific condition, when magnetic field strength will exceed the strength of radio frequency, so in that condition protons will be aligned in the direction of magnetic field, and this is known as resonance. When this resonance in the protons takes place, then receiver coil will receive the signal and it will pass the signal towards the amplifier. Then amplifier will amplify the signal and it will go towards the detector, and finally detector will pass the signal towards the recorder, and ultimately we will get the NMR signal. So this is the details about instrumentation and the working of NMR spectroscopy.
So we have seen how this is working. So let's see the applications of NMR spectroscopy. NMR spectroscopy is used in structural elucidation of the compound, and it will also count the number of peaks which are recorded and number of types of protons which are in this compound. NMR spectroscopy helps in the determination of optical purity, and it is also used in quantitative analysis also. Let's see the last and final one, which is electrophoresis. This electrophoresis is movement of charged particles under the influence of this electric field. When we are applying any electricity, they will be movement of this charged particles. So mostly we will use this for proteins, DNA, to recognize the proteins which are in compound, and it was also used in DNA and RNA analysis. So let's see how this will work. This technique is nothing but we will apply to a liquid medium, we will apply this electric field. So when we are applying this electric field, the liquid medium will migrate to the electrodes of the opposite charge. For example, cations are positively charged, so they will migrate to negatively charged electrodes. Okay, and anions which are negatively charged, they will migrate to positively charged anode electrode. So this force is like this migration depends on the charge of the molecule. So if there is greater the charge, then migration will be very forced. Like for example, if if the anion have very strong negatively charged, then it will travel very fast. Okay, but if it is having very low, that will travel very slow. Okay, so let's see the types of electrophoresis. We have different types of electrophoresis, like free electrophoresis, zone electrophoresis, capillary electrophoresis, gel electrophoresis. So let's see what is free electrophoresis. As this is free electrophoresis, we won't keep any liquid medium or any medium to this. Okay, we will just keep the plate, and that we will apply to that plate, we will apply this electric field. Then the cathodes and cations and anions will travel. And zone electrophoresis. In this zone electrophoresis is also known as electrochromatography, in which separation is carried out by using a medium like paper or gel. Okay, so we will take paper or gel and we will apply the compounds, and with that we this cations and anions will get separated, and also we will apply this electric field. And let's see the capillary electrophoresis through a video.
Capillary electrophoresis is a method for separating and identifying fragments of fluorescently labeled DNA by passing them through very thin polymer-filled capillaries. The use of capillaries instead of a conventional slab gel makes it possible to apply a much stronger electric field, resulting in faster separation and higher throughput. The process features single base resolution and requires small sample quantities. Applications include STR analysis and Sanger sequencing, as well as many other everyday applications used by life science and forensic laboratories. Here's how it works. First, fluorescently labeled DNA samples are prepared for electrophoresis and loaded into the CE instrument. The capillary array is filled with polymer and placed into the sample wells. When the capillary array enters the wells, an electric current is formed and the DNA fragments are moved electrokinetically into the capillaries. Next, the ends of the capillary array are inserted into the cathode and anode buffer. The instrument applies a specific voltage and temperature to control sample migration through the capillary array. The fragments are separated by size, with the smallest fragments moving fastest. The final step is sample detection. As the fluorescently labeled fragments move past the detection window, a laser excites the fluorescent dyes. The emitted light is spectrally separated and recorded by a CCD camera. The CCD camera records the fluorescent signature of each fragment, as well as the elapsed time for the fragment to migrate through the polymer. The raw data is sent to the collection software in the form of a peak, and is now ready for analysis using your choice of software program. This animation is brought to you by Spectrum Compact CE System.
This kind of an image is not new to us, especially for people who love forensic science. But what exactly is this? It's an image of the separated DNA fragments on a piece of gel. And the process of separating them is called gel electrophoresis. Doesn't this sound very difficult? Separating DNA fragments on a piece of gel? But actually, the process of gel electrophoresis is not very difficult. In fact, it's quite easy. Let's have a look at how the process works and also the principle behind it. Let's begin with the definition first. Gel electrophoresis is a technique to separate the different DNA molecules based on their sizes. Can you guess why is it called gel electrophoresis? Okay, let me help you with this. This separation technique is based on the movement of charged molecules when exposed to an electrical field. And this movement occurs in a gel medium. Hence, it's known as gel electrophoresis. Now let's understand the process step by step. But before that, Since the term charge is used by us here, can you guess the charge present on a DNA molecule? Let me tell you that a DNA is negatively charged. And this is due to the presence of phosphate groups. These phosphate being negatively charged and present in the backbone of the DNA double helix impart a negative charge to the complete molecule. And what is the gel used? Interestingly, the gel which is generally used for the DNA is agarose gel, and it's obtained from the seaweeds. Now getting back to our process. We said that the DNA molecules get separated on the basis of their sizes. So if we have a sample of DNA which is containing fragments formed with the activity of restriction enzymes, then separating the fragments based on the difference in their sizes would be possible with gel electrophoresis. Let's have a look at this in detail. To understand the principle and working, let's take the analogy of a sieve. We know that if a mixture of different sized particles is poured onto a sieve, then only the particles smaller in size than the pores will escape, while those bigger in size will be retained back. Here, the size of the particles is the only parameter used for the separation. Same is the case with DNA fragments. When our DNA sample containing various size fragments is loaded onto a gel, then the charge applied across the gel helps in the mobility of these fragments. However, not all fragments move at the same pace. Those which are smaller move ahead, and those which are larger in size find it difficult to move. To understand this better, let's zoom into the gel that is prepared for the experiment. Here, at the microscopic level, we find that the gel appears to be a mesh-like structure and the pore size is nearly constant throughout. So now imagine what will happen if different sized DNA fragments are made to pass through these pores. It's obvious that the small size fragments will escape the pores faster, while the larger fragments will find it difficult to come out. They will take a lot of time to cross the smaller pores and thus will lag behind. This is how we can understand the principle of separation of the DNA fragments based on their sizes. Now let's see how the procedure works. The first requirement is the complete setup. This will include the casting tray, gel, a comb for making wells in the gel, electric supply, and most importantly, DNA sample with different sized DNA fragments. Here's an illustration of the casting tray containing the gel in which we will load the DNA mixture. We usually opt for a comb to form well-like structures into the gel. And why do we do that? Because it's in these wells in the agarose gel that we load the DNA mixture. Now tell me, where should the wells be formed? At the ends or at the center? Think about it. We know that the DNA fragments, being negatively charged, will travel from a point of negative charge to the point of positive charge. Thus, we need to form the wells to load the DNA samples near the negative terminal or the cathode to be precise, so that the DNA will move towards the positively charged anode. This movement of the DNA molecule will be promoted by the electric field. But we cannot track the movement of the DNA in the gel as it's colorless. For this, we use a colored loading dye to track the movement of the DNA. The mixture of the DNA samples with the colored loading dye is now all set to get into the gel. But before loading the DNA mixture, we will fill the apparatus with a buffer as shown. The buffer is used to provide better conductivity of electricity. Next, we will load the DNA mixture into the wells and we're ready to turn on the power supply. Once the power supply is turned on, the DNA mixture will move through the gel, which can be tracked with the help of the loading dye. The loading dye is selected in such a way that it travels a bit faster than the DNA segments present in the mixture. And why is that so? That's because we want the loading dye to reach the terminal end faster than the DNA. Once the dye reaches the anode, we get an indication that the DNA must have reached somewhere near and thus the power supply has to be turned off. Moving ahead with the next step now. We know that the DNA molecules have got separated. But can we really see them? No, that's not possible. So how will that be possible now? How do we observe the separated segments? To observe the DNA molecules, we treat the agarose gel with ethidium bromide solution. The major reason for using ethidium bromide is that it easily binds to the DNA molecules, and when the agarose gel containing the DNA is observed under ultraviolet light, bright orange colored bands are clearly seen. These are nothing but the bands of the DNA. The separation has been successful based on the size of the DNA fragments. The larger molecule are the ones found here, which means these are the ones that moved slowly. On the other hand, the smaller molecules which moved faster are the ones spotted here. Now, how do we know the size of any DNA fragment? The bands obtained are compared with a standard chart known as the DNA ladders. On comparing the positions of the bands with the ones from the chart, we can easily make out the length of the fragments obtained. Once we know the lengths of the fragments, we can easily identify the desired DNA. Now, the desired DNA fragment can be first manually cut out from the agarose gel and then extracted. This process is called elution. The extraction of the DNA is done in such a way that it can be used for further downstream processing. This was the simple explanation of how any gel electrophoresis technique works. It's considered as one of the very important techniques used in recombinant DNA technology. To learn more about such interesting processes, stay tuned to our channel and do not forget to subscribe. Happy learning!
So this is electrophoresis. By this, we have completed the fifth unit. Thank you, everyone.